Over 400 years ago, during the Qing dynasty, the Kongxi Emperor sought to prevent smallpox after his father's death. For thousands of years, smallpox had ravaged the globe without answer. But Chinese doctors were developing an early form of immunization. They were exposing healthy people to small amounts of variola minor, a milder smallpox strain. Under the emperor's rule, the developing practice became widespread, and his people slowly gained protection from the disease. This marked the first known immunization campaign and led the foundation for vaccine development. In the centuries since, public health has been safeguarded by similar logic, that the immune system can learn to prevent disease. Today, thanks to advances in microbiology, immunology, and healthcare, we can effectively train the immune system through vaccination. Vaccines are undeniably among our greatest medical achievements, saving countless lives. And while today's vaccines are no doubt the product of global research efforts, some of the most significant strides in vaccinology trace back to McMaster University. From pioneering critical research tools to leading innovation in inhaled aerosol vaccines, this is the history of vaccinology at McMaster. It's the early 1970s, and scientists around the world are looking for better ways to study viruses and develop vaccines. In the Netherlands, a Canadian researcher named Frank Graham helps create a groundbreaking tool, a special type of human cell that can grow viruses safely in the lab. These cells, known as HEK293 cells, allowed scientists to test new vaccines quickly and efficiently. In fact, decades later, these same cells will come to play a key role in developing the first COVID-19 vaccines. In 1975, Graham brought his research to McMaster, where he established a lab in the university's life sciences building. There he continued to refine and expand the cell line's potential. This work helped lay the foundation for vaccine development, both at McMaster and across the world. During the 1990s, Graham's continued research on HEK293 cells led to a new McMaster-made vaccine, developed in partnership with biology professor Ludwig Previk. Indeed, Graham engineered adenovirus to make it a convenient vector for introducing genes into mammalian cells, and Previk developed a vaccine against rabies using this technology. After rigorous testing, the vaccine was acquired by the Ontario Ministry of Natural Resources and Forestry and is now widely used across Canada and the United States to help control rabies in wildlife. This vaccine is not administered by traditional means. Instead, it is packaged into bite-sized baked pellets that are scented, flavored, and consumed by animals. Hundreds of thousands of these pellets are routinely dropped by airplane into areas rich in wildlife, helping at-risk animals gain protection from the otherwise deadly virus. The foundation, laid by Graham and his colleagues, inspired a new generation of McMaster scientists to pursue vaccinology at the university. Through collaborations between McMaster's biomedical scientists, clinical infectious disease specialists, respirologists, and chemical engineers, the university became a nexus for research into the development and testing of novel tuberculosis vaccine candidates. In the early 2000s, McMaster University, under the leadership of Professors Zhou Xing and Fiona Smale, developed a groundbreaking tuberculosis booster. This new vaccine offered enhanced protection for those who had already received the standard BCG tuberculosis vaccine. Human trials showed that McMaster's innovation could effectively reactivate the immune defense that tends to fade over time after initial vaccination. In time, Xing and Smale would develop a new tuberculosis vaccine candidate that would be administered via inhalation instead of injection. Unbeknownst to them, this new inhaled vaccine platform would position McMaster at the front lines of the response efforts to one of the greatest health emergencies of the past century. Meanwhile, McMaster Professor Karen Mossman was working with her team to study virus-host responses in a specialized facility, work that would allow them to make a major scientific contribution 2 decades later, when our world as we knew it would change.
Chinese health authorities are still working to identify the virus behind a pneumonia outbreak in the central city of Wuhan.
Fast forward to 2019, when a new infectious disease emerged. Clusters of people began experiencing a pneumonia-like illness that would not respond to standard treatments. Public health authorities identified the mysterious pathogen to be a novel coronavirus, which caused a disease that would come to be known as COVID-19. Quickly it spread across borders, across oceans, and within weeks, the new virus was confirmed in Canada.
As you're speaking, and as we're both sitting here, the World Health Organization has just declared that this is a pandemic. So this is their declaration.
At McMaster, a lot changed. Students and staff were sent home, many operations were paused, and campus was effectively closed. But a lot persisted too. In particular, the university's vaccinology work, now more critical than ever. McMaster researchers in Mossman's group played a key role on the small team that successfully isolated and grew copies of the virus responsible for COVID-19, creating a source of virus for study as the world raced to develop vaccines and treatments. Meanwhile, in another McMaster lab, Zing and Smale, now with colleagues Matthew Miller and Brian Lichty, quickly pivoted their inhaled tuberculosis vaccine platform to face the new threat.
Researchers here at McMaster University are developing what they call the next generation of the COVID-19 vaccine, and this one will be needle-free.
By May 2020, less than 2 months after the pandemic declaration, the McMaster research team had secured millions in funding to develop inhaled aerosol COVID-19 vaccines. Importantly, pivoting from tuberculosis to COVID-19 in short order showed considerable versatility in the university's inhaled vaccine platform, leaving researchers certain that it can help us face future threats. A revolutionary cell line, rabies vaccines, novel tuberculosis boosters, and an inhaled aerosol vaccine platform. These milestones dot McMaster's vaccinology timeline, but hardly tell the full story. Indeed, McMaster experts have also explored universal influenza vaccines, made advancements in passive immunization strategies, developed biotechnologies that may soon lead to new vaccines for cancer, and developed technologies to better stabilize vaccines. McMaster experts have informed government and public health groups on vaccine policy, led important clinical trials activity, studied and supported vaccine access, safety, and equity, and synthesized immense evidence that has helped inform vaccine-related decision-making across the globe. Over the past 50 years, McMaster has made critical contributions to the field of vaccinology.
But that's enough about the past. The future starts now.